All right, welcome back to our discussion of metabolism. Today we're going to move into concept 8.3, ATP powered cellular work by coupling exergonic reactions to endergonic reactions. So in the cell, we need to do three main types of work. Either chemical work, where we're making things, transport work, where we're moving things across the membrane, or mechanical work where we're either moving the cell, moving flagella, um, contracting a cell, any type of actual physical movement. In order to do this work, we need to couple that to the cell's energy resources. And so, since most of these various forms of work are going to involve an energonic reaction, what we do is we couple it to an exergonic process in order to drive that forward. So we essentially harvest some of the energy from this reaction, which has a negative delta G and is spontaneous, in order to make this one happen. And by far the most common is those mediated by the hydrolysis of ATP. So just to remind us, ATP is a nucleotide, just like the ones that build our DNA and build our RNA. So here we have a ribose ring. Notice that it is not a deoxy form that we're using here, so it's more of an RNA form. Here's the nitrogenous base, and we have these three phosphate groups added on to the 5' carbon. Now, it's these phosphates that really are where we get that energy from. So these phosphates are very electronegative. They have negative charges associated with them. They don't want to be attached together. With just a little bit of nudging, you can get them to pop off one of those phosphates. And it does this by adding water. So this is um, hydrolysis reaction, just like we've seen over and over again. Now, when this happens, there is a release of energy from this. So this has a lot of stored energy, a lot of potential chemical energy. And when it's released here, we can do things with that. So the bonds between phosphate groups of ATP's tail can be broken through hydrolysis. That energy is released from ATP when the terminal phosphate bond is broken, and this energy release comes from the chemical change to a state that has lower free energy, and also is favoring entropy, right? So releasing that phosphate is more disordered. So the three types of work that we can do is mechanical, transport, and chemical. This is can all be powered by hydrolysis of ATP. In the cell, the energy from this exergonic reaction, this ATP hydrolysis, can be coupled to an endergonic reaction, something that requires energy. Overall, the coupled reactions are still exergonic, right? So ATP drives these reactions, usually does so by phosphorylation. So we transfer that phosphate group from ATP onto something else, such as the reactant or sometimes the enzyme. In this process, we develop something that is called a phosphorylated intermediate. This is also a stored bit of energy, and the phosphorylation in this phosphorylated intermediate can also be released, harvesting even a little bit more energy. So let's take one reaction that our body carries out. So here we have glutamic acid. It's one of our amino acids. And if we add a couple ammonia onto it, we can turn glutamic acid into glutamine, another one of our amino acids. This has a positive delta G by 3.4 kilocalories per mole. So what we have to do is we have to couple this with the phosphorylation of glutamic acid. So if we take glutamic acid, add hydrolyze this ATP, releasing that phosphate, putting that phosphate onto the glutamic acid, creating this phosphorylated intermediate in ADP. This can, the release of that phosphate has enough energy in order to allow it essentially trading positions with this ammonia. So here we transfer it onto this molecule, we create an energized phosphorylated intermediate. This phosphorylated intermediate can then carry out the original reaction that we wanted and have the energy to proceed. So if you look at the net energetics of this, ATP to ADP is minus 7.3 kilocalories. This original reaction was plus 3.4 kilocalories per mole. This gives us a net negative delta G of 3.9 kilocalories per mole. So this reaction is now favorable because we've coupled it. Here's an example of various ways that we can do work. So here's a transport work. This is active transport. So we have this protein in the membrane. It's going to bind something, and it's going to move it up its concentration gradient. So it's not something that's favored. Here, what we can do is phosphorylate the protein, creating a phosphorylated intermediate, and then use the release of that phosphate to drive a conformational change, which allows that solute to be open to the other side. Down here, we have vesicles walking along a cytoskeletal tract. This is microtubules. And this motor protein is driven by ATP. And so in the process of phosphorylating the motor protein and then releasing the phosphate, it causes conformational changes in the protein, just like it does up here, and it causes it to walk forward. And it also opens binding and then closes binding to microtubules. So this is kind of a multi-step process. Ultimately, this ATP that we hydrolyze and use can always be recovered by the food that we eat. So as we break down our food through catabolism, we can regenerate that ATP. We can then use that ATP to do work. So ATP is this guy here. It's just simply a nucleotide with 3 phosphates. These 3 phosphates, the bonds want to be broken. We can utilize that activity to couple reactions that are not favored to reactions that are favored, creating an overall reaction that still has a negative delta G. And oftentimes we do this through phosphorylation, either of one of the reactants or through the enzyme itself. And doing this, we can not only do enzymatic reactions, but also transport and move things throughout the cell.